So after students inoculate all those tests in exercise 16 to try to help identify their gram-negative, students may think, okay, if I was really doing this, say, in a hospital lab, do they really inoculate all these test tubes? Well, the answer is probably not. So I like to show this picture. This is an API 20 test strip, and these are especially useful in determining what some unknown gram-negative rod-shaped organism is. Okay, so they have different test strips, say for gram-positive cocci, but this is probably the most commonly used test strip. And the way these work, when you first get them, they look like this. Up at the top here, there's these little plastic wells. They actually have an opening at the top. It's kind of hard to see that. And you can see some of them have some reagents in here. Well, they all have something in them. They have reagents in there that are dried down, basically. So the organism that you're trying to identify, you suspend it in some sterile saline. And then you put a drop or a couple drops into each of the wells. You use a pasture pipette probably. And that lays down flat. You incubate it for 24 hours. And then for some of the wells, you need to add some kind of reagent, just like you did in class, like you added Kovacs reagent for the indol test. You add a drop of the reagent on top. And then you look at the color changes. So these are 4 different organisms inoculated into an API 20 test strip. And the instructions tell you what color changes to look for as to tell whether you have a positive or negative test. Now, hopefully some of these look a little bit familiar now. Let's see, CIT, this is a citrate test. So blue here, that's going to be a positive, and green is negative down here. H2S, we're going to test for hydrogen sulfide today. URE, I believe that's a urea test. Here's a VP test down here, Voges-Proskauer. This is hard to read. It's GEL. It's actually a gelatinase test. Typically, we do that, but you wouldn't have done that this semester. All the rest of these tubes here are testing for sugar utilization. So we've got glucose, mannitol, arabinose. I don't even know what they all are. But here we have an organism that is not fermenting any of those sugars. And down here, here's an organism that is a little bit more flexible with their metabolism and is fermenting all the different sugars. So there's a big variation in how gram-negatives, especially which sugars they can utilize and which ones they can't. So you score each one of the tests, and according to the instructions, you assign your organism a number. You feed the number into the website, and it will tell you with a certain degree of certainty what your organism is. So really nice and easy. You can do a bunch of tests all at once. You get your results back within 24 hours. These are a little bit pricey, so that's why we don't do it in lab. And when you do the tests one at a time, then you have a chance to really kind of understand what's going on. Okay, so I just like to show you that, just so you know that it's out there. With Lab 17 involves only inoculating 2 more tests. And both of these tests are considered multiple test media. And that means you're testing for several different things within a single test tube. So again, that's more efficient. You use less media, you get more information. Sometimes it makes the tests a little bit more difficult to read. Sometimes one result might actually mask another result. Okay, I see my picture turned sideways. I don't know how to keep that from happening. Sorry about that. I have some other pictures here that I hopefully will be better. But the first thing you're going to inoculate today is Kligler's iron auger. So we usually abbreviate that KIA. Okay, and within this one test tube, we're going to test for 2 things: sugar utilization, and hydrogen sulfide production. Okay, and this, again, my picture is bad here. These are inoculated into test tubes that are a little bit larger. This is solid media here. I'll talk about how to inoculate those in a minute. What's in this media? If you understand what's in the media, then it's a little bit easier to understand what we're testing for and what results we're looking for. Okay, so this KIA media contains number 1, glucose, and number 2, lactose. We actually have about 10 times more lactose than glucose. So essentially, we're giving the organism just a little smidge of glucose, and then we want to see what happens when that glucose runs out. Can the organism use lactose, or does it have to start using some proteins in the medium to metabolize for energy instead? Okay, so anytime we're looking for sugar utilization, we also need a pH indicator. We're actually using the same pH indicator that we used for the urea test. Colors look a little bit different here. It's phenol red. So if you remember, with the urea test, it was kind of a light tangerine orange to begin with. This is a little darker orange to begin with. But still, we're looking for a yellow color change today because we're really looking for mostly acidic end products, but if we got more alkaline, then we would get it to be even more, a little bit more red than what it is already. And actually, we could see either yellow or red. Okay, we're kind of looking for both. Again, if the organism uses lactose, then we're going to get some yellow. If it can't use the lactose and the glucose has run out, then we're going to expect to see some red. All right, so we'll talk about that in a minute. The other thing we're looking for here is hydrogen sulfide production. So we have to give the organism a source of sulfur, and that's in the form of sodium thiosulfate. Okay. And we need an indicator for hydrogen sulfide production, and that's ferric ammonium citrate. Again, that's an indicator for hydrogen sulfide. Okay, hydrogen sulfide is a colorless gas that's produced as a result of the organism doing some anaerobic respiration. Okay, so typically in the past, when we've tested for gas production, we've had a liquid media and we've had a Durham tube in there to catch the gas bubble. Well, today we've got a slant, we've got solid media, but this indicator, ferric ammonium citrate, that's a source of iron. So if the organism produces hydrogen sulfide, remember it's the bacteria that's doing that, this ferric ammonium citrate, again this source of iron, combines with the hydrogen sulfide and forms a black precipitate, iron sulfide. Okay, black precipitate. Okay, so if the organism is producing hydrogen sulfide, we get a black color change. Okay, and that's always going to be pretty obvious. Okay, so there's a specific way that I want you to inoculate your KIA. Like I said, this is a slant. So to inoculate, we're only using our needle with Lab 17. So to inoculate both tests, again, you're going to use your needle and you're going to touch some of your assigned organism. You're going to first stab down in the auger, and then secondly, you're going to streak the surface of the slant. Okay, so you're going to stab down in, come straight back up with your needle, and then streak the surface of the slant. Okay, notice that I've stabbed kind of down the lower portion of the slant. If you stabbed up here, you would likely have a large portion of the tube where nothing was growing. Okay, so we really want it to grow throughout the tube, so streak it down kind of at the lower portion there is always going to be better. And the reason we want you to stab first is because, let me go ahead and write this here, stab first because you're going to have, because you want more of your inoculum in the bottom of the tube, where the organism is going to grow more slowly. Okay, there's less oxygen down there. Okay, so it's going to take longer to grow. Get your heaviest inoculum down at the bottom, and then if you just have a few cells on the surface, that's fine. That'll be enough. They have access to oxygen, and they're going to grow really quickly. Okay, so you don't need to re-dip into your stock culture. Just stab and then streak with whichever's left on your needle. Okay. Whenever we are reading our results, we always read the results of a KIA test in two parts. We're going to read the slant portion separate from the bottom of the tube or the butt of the tube. Okay, and if we have a yellow color change, that means it's an acidic pH, and we're going to abbreviate that A. If we have a red color change, then that's an alkaline pH, and we're going to abbreviate that K. Okay, and you always want to read the results after 24 hours. Whenever you're looking at sugar utilization, you have to be careful about the timing. Again, if you let it go too long, or if you remember back when we were talking about fermentation tubes, if you let it go too long, we could get alkaline reversion. Okay, and then your results are not going to be accurate. Okay. All right, let's see, how much room did I give myself? Okay, I got another page. All right, so let's look at what the results could possibly be. You may have a tube where, so again, this is after incubation, a tube that's yellow on the slant and yellow on the butt. All right, so we would read that sugar utilization A over A, and we would interpret that as the organism utilizes or ferments glucose and lactose both. Okay, so even when the glucose runs out, we still get acidic end products. Your organism may be red on the surface and red on the butt, so we would call that K over K. All right, and we interpret that as neither lactose nor glucose fermented or utilized. Okay, and a couple of our organisms will look like that, and that's a really kind of distinct result. Lastly, in terms of color change, you may have red on the surface and yellow on the butt. So we would score it K over A. And we're going to interpret that as the organism only is using glucose. Okay. Now, the reason is, if you were to look at these tubes maybe after a couple hours, the whole thing would be yellow. But once that glucose runs out, we start to get some alkaline reversion. Okay, and so the organisms, again, start using the proteins. And remember, when they're using proteins, they're having alkaline end products. So on the surface, where the organisms are growing more quickly, it's going to burn through that glucose more quickly, and we get that reversion more quickly. Eventually, that reversion would move all the way down to the butt. But again, at the time that we read these, about 24 hours, there's still some glucose down here because the organisms are, again, burning through that a lot more slowly because they don't have access to oxygen. All right, so eventually the whole thing would be red, again, once the glucose runs out and the organism can't use lactose. Okay, remember, in addition, we're looking for hydrogen sulfide production. So if we have any black anywhere on the tube, then that's an organism that produces hydrogen sulfide. You will only see black in acidic areas. Okay, so if you've got something like this, red and black, you can assume that it's acidic underneath there. But it might be a little bit more subtle than this. You might have just a little bit black. One of our organisms kind of only weakly produces it with this test. So that will be pretty obvious. Okay. One last thing, your book mentions using this test to determine the fermentation pathway. And the way they say to do that is to look for gas production. So if you see some big cracks in the agar like this, or sometimes that solid media is totally pushed up from the bottom of the tube, then the book mentions that that organism is probably using a mixed acid fermentation, because we typically get more gas as a byproduct of that fermentation pathway than if organisms use the 2,3-butanediol pathway. The problem is that's not a foolproof thing, and it's kind of hard for us to know what's a lot of gas, what's a little gas. So I say, do a methyl red or a VP test to determine fermentation pathway, and don't rely on a KIA for that. Okay, but it's something that you can make a note of. Some of the organisms, again, make a lot of gas, and that may be helpful. Okay, so here's just a few pictures of some inoculated KIA tubes. Here, this one over here, these have been inoculated and incubated. Okay, here's what that K over K looks like. Here we have K over A plus H2S. We got a little bit of everything there. It's very nice. Here's that A over A organism that's using both glucose and lactose. Okay, the SIM test is not nearly as complicated as the KIA, so that's good news. Once again, though, multiple test media, and there's 3 different things we're testing for. SIM is an acronym for those 3 things. So the S stands for, sulfide, actually hydrogen sulfide production. Okay, so that's redundant. This is another way, besides the KIA, to test for hydrogen sulfide. It's a little bit more sensitive than the KIA. So one of our organisms, sometimes we don't see hydrogen sulfide production with the KIA, but it always shows up in the SIM. So if this is a really important result, you should do a SIM instead of a KIA or in addition to a KIA. Okay, but it's detected the same way. It's got that ferric ammonium citrate in there, looking for a black color change for a positive hydrogen sulfide production test. The I stands for indole. So this is another repeat, right? We also tested for this as part of the IMVIC And really, it's the same way. We're reading it. We're going to have to add Kovacs, about 5 drops. And remember to read that. We're looking for a red ring sitting on the top of the broth. Okay. The only thing new here with the sim test is a motility test. Okay, and there's actually a lower percentage of agar in this medium. It's really kind of semi-solid. Okay, so that's to allow organisms that can move around, makes them able to actually do that. And remember, what determines whether an organism is motile or not? Do they have flagella? Okay, if they do, then they're motile. If they don't, then they're not. All right. So most of our organisms are motile, but there's a few of them that aren't. Okay, so to inoculate the sim test, it kind of looks something like this. Again, to inoculate, you're going to use your needle and streak maybe 3/4, 2/3 of the way down and try to come right back up with a steady hand. No need to streak on the surface there. Your needle would kind of smush right down in there because it's again kind of not as solid as, say, the KIA is. So again, just about 3/4 of the way down to inoculate. We're going to incubate that. And then, let's see, you already know how to read the hydrogen sulfide and the indole. For the motility, if you have growth only around the stab, then guess what? That organism is negative for motility. Or you could say non-motile. Okay, and a couple organisms that will be pretty clear. If you have growth around the stab, but it's also cloudy, or you might not even be able to see the stab anymore, actually, then that's an organism that's positive for motility. I want you to understand that this can be very difficult to read because some of our organisms don't grow very well in the sim medium. So to tell, is this cloudy, is it not cloudy, it's kind of subjective for some of the organisms. So I tell students I would never want to make a call based on what my organism is based on motility. Now, there's a couple organisms where it's pretty clear, but I would say that's a pretty tough call. That's one of the reasons that I like to also do a wet mount where actually seeing the organisms alive, we can watch them and see if they're moving in distinct ways or whether they're just flowing through the medium. So if we have time to do that, we will. I'm not sure if we will this semester. Okay, so remember, we're also looking for this black color change. So if you have an organism that's a hydrogen sulfide producer, your tube's going to look like this. And then, well, how do you determine the motility if it's positive for hydrogen sulfide? Well, it's pretty difficult. But if you've got hydrogen sulfide diffusing throughout that whole tube, then it's probably going to be positive for motility as well. But again, that makes it difficult to call for sure. Okay, so here's just a couple of examples. Here it looks like an uninoculated sim tube. Here's one that's positive for hydrogen sulfide. Remember, you have to add Kovacs reagent to read the indole, and I'm not sure if that's happened with this tube or not. But that's the KIA and sim tests, and that's all the new things we have for you to inoculate. So have a good day.